Thanks, John, for organizing this wonderful workshop. And yeah, I'd like to thank all the speakers to share their fantastic works. And here, I'm Jiyoung Kwan, a postdoc in TechChip Lab. And yeah, here we are investigating the cell morphologies and the protein distributions inside the minimal cell by using imaging techniques. And especially, we are focusing on the cell division process recently, based on the recent simulation results from our collaborator, Lucy Schultengroup. So yeah, I think that we all are quite familiar to the development of these minimal cell lines and the difference between those synthetic cells. So here we have scene 1.0, the first synthetic minimal cell, and scene 3.0, the first synthetic minimal cells, and the scene 3A, the minimal cell with a better morphology. And here we have one more synthetic cell lines called scene 3B, in which we have a genetic platform that allows us to express extra copy of the genes. And the big difference between the scene 3.0 and scene 3A is a much better homogeneous cell size distribution in the scene A. And we all know that scene 3A has FTSC and SEPF genes adding to the scene 3.0. And those two are known to play some roles in the cell division. So maybe they also play some roles in the cell division of the minimal cells. And one of our collaborators, the resistant Schultengroup in UIUC, tries to do the whole cell simulation based on the some experimental measurement. And they provided wonderful measurables such as mRNA half-life or the concentration of very important macromolecules. And one of interesting features is their estimation about the cell growth rate and the cell volumes during the whole life cycles. And the conclusion is that the cells may be divided equally in terms of their volumes or surface areas. So as an imaging group, our goal is quite straightforward. First one is that investigate the subcellular structures if they are exist inside the cells, and investigate the distribution of some important proteins such as PTSG or FSC. And based on previous simulation results, we'd like to visualize the dividing cells directly by using some imaging techniques. And one of our goals is observe the whole cell dynamics by using the light microscopes while the cells are alive. But yeah, we couldn't reach this region yet. So yeah, this goal is still remains as a future works. So for the first part, we started to visualize some proteins inside the cells. And we all know that the minimal cells has a size less than 1 micrometer. So directly to visualize the proteins, we need to utilize some super resolution techniques. So here I have used the storm microscope, and the fluorophore was labeled on this black text. And yeah, I applied the storm technique on these Alexa 647 dyes. So these two are the wide field images of MF, the fluorescence protein tag on the proteins, and the Alexa 647 immunofluorescently labeled on the black tag. So from the wide field images, we confirmed that the IF works fine. And from this Alexa 647 fluorophore, I could get some single molecule localization images. And here I'm gonna show you some more example of these 2D storm images or the 3D distribution of the FTSC proteins. So from those images, I could say maybe the PTSG proteins are localized on the membranes. And yeah, mainly they are clusterized, and we can see some potential membranes without the PTSG proteins. So yeah, it could be their natural behavior, or our sample preparation step perturbs their localization, like such as permeabilization steps. And yeah, without a clear reference structure, yeah, it's a bit hard to interpret further details of this distribution of PTSG proteins. And our next target was the FTSC proteins, and we previous work clearly showed that FTSC forms a ring-like structure during in the dividing cells. And here we could observe some half ring-like structures, but yeah, so far we didn't see any clear ring-like structure from the FTSC proteins. So again, yeah, maybe it's hard to interpret the super resolution images without a clear structure. So for the reference structure, I'd like to introduce, I introduced the membrane probe called SPPIOC, and uh, to investigate if this dye can be applied for the super resolution imaging, yeah, I checked its blinking behavior, and yeah, I could get some super resolution imaging from this membrane probe. And here's a colocalized 2D super resolution imaging of FTSC protein and the membrane dyes. So it seems, yeah, this FTSC is localized on the membrane, but still it's very hard to, yeah, the structure is not that smooth, but still clusterized. And yeah, even with some contours, I could get a bit clearer membrane structure, but still it was very hard to get a clear, very clear resolution images from those two structures together. And with a additional staining on the DNA, I could see a potential dividing cells. So here I can show you some different type of images, and here the cyan ones marks the well colocalized signals between DNA and membrane and cytosol channels, which could be the intact minimal cells. And I could see some just aggregation of membrane probes. And also some cells are showing clear DNA and membrane signals, but they are absent of the cytosol signals, and I still don't know what they are. And here, the white one, it has clearly separated DNA and cytosol structure, but yeah, the membrane looks still continuous. So this could be a dividing cells, and the volume of divided DNA or cytosol looks pretty equivalent as predicted from the simulation. So to investigate this cell division process, we changed the membrane probe for better visualization, and we utilize other microscopic technique called air scan to get better images. So here's some example image from the air scan. So if you compare this merged image to the previous wide field image, now we can see much clearer membranes and the DNAs, as well as the FTC distribution inside the membranes. So from these air scan images, I picked some interesting features. So the first one is these single cells. So now we can see the very clear membranes, and they are well encapsulate the FTSC and the DNA signals. And there are some single cells that are very elongated and have the localized FTSC signals in the middle of the cells. And there are also potential dividing cells. They could be just two adjacent cells, but when I precisely look at the distribution of DNA and the FTSC, those two contents are linked, although they showed some development of the membrane between two cells. And here is a movie that I like much, which is the 3D image of these potential dividing cells. So we have very clear outer membranes, but there is still a weakly developed membranes between two cytotonic contents. And although the simulation estimated the symmetric cell division, I also could see some budding-like behavior. So I'm working on the quantification of these potential dividing cells to see whether the symmetric division is measured or not. And from the single cells, I'm also doing some statistics to get the size measurement as well as the ellipticity. And I'm also working on the distribution of the FTSC to characterize the status of the cells. And yeah, I look at the correlation between the ellipticity and the diameter. And my idea was that if the cells are growing, maybe it's more elongated, and the FTSC has more developed structure inside in the middle of the cells, but so far I couldn't see any correlation between these three phenotypes. And finally, here is my very hypothetical morphology changes during the cell division. So when the cells are getting bigger, the FTSC signal starts to concentrate in the middle of the cells, and it's getting bigger and bigger, and it's elongated, and you can see the very well developed FTSC bridge in the middle of the cells. And then finally, the membrane starts to shrink into the middle of the cells. Then the inner membrane structure, not inner membrane. The membrane structure between the cells are clearly developed, but still their contents are linked between the cells. And then finally, the membranes are fully separated after the cell division. So, but still, yeah, we don't have any further support to claim this hypothetical model. So we are very interested in the any kind of clue for some proteins whose expression level might be changed during the cell cycle or something like that inside the minimal cells. And yeah, I still need to do some quantitative analysis, and it's pretty hard to interpret the FTSC distribution clearly. So yeah, I'm still thinking about it. And yeah, we are also interested in the high resolution imaging on the DNA structure, because yeah, sometimes I could see some circular DNA structure, and sometimes it's very heterogeneous. So yeah, I'm just very interested in the actual DNA distribution inside the minimal cells. And also we are it would be great if we can do the similar things with the live minimal cells. And yeah, finally, I could see very frequently these empty and abnormal membrane structures inside the cells. So it would be great if we can figure out what they are, yeah, from the image results. So yeah, thank you so much for your attention. And yeah, this is one of collaborate. We are collaborating with these many groups for this talk. So thank you again, and I'm happy to take any questions.
Ji-Won, I'm stunned by your images. Questions?
Yeah.
if I can lead off, a few slides ago, there was
Mhm.
one of the cells that you showed. Keep going back. There.
Yeah.
So sort of in the middle of the slide, there is a cell that looks like it has a plane of FTS. So you've got what looks to be cells that are dividing, and then another large cell that has a central plane of FTSZ.
Yeah, like here.
Yeah. Talk to me about that one, please.
I have no idea. So yeah, my brief idea is that the FTSC may be built up when the cells are close to be divided. But yeah, still I could see very for the potential dividing cells, this behavior is very common. So here, even you can see the FTSC is localized at the opposite polar of the dividing cells. So so far, yeah, I couldn't correlate any I couldn't correlate the distribution of FTSC and the morphology of the membrane clearly. So yeah, unfortunately, I have no answer for your question.
So we just need cells that are like 10 times larger. Other questions for Ji-woong?
May I ask question?
Of course. Go ahead.
My name is
yourself, please.
I'm Daisuke Shiomi from Japan. I'm sorry, my camera does not work. So my lab is working the similar thing. We are observing the GFP FTSZ fusion, and we see the similar mid-cell localization of FTSZ. So my question is, does FTSZ localization is dependent on FTSA or CEPF or other proteins?
Oh, so far, yeah. So far, I have not visualized the FTSA and FTSC together, and I have done observed the CEPF. So those are future works, not listed on the the PPT. But I'm also very interested in the correlation between the FTSC and FTSA because it's well known that FTSA actually tethers the FTSC proteins on the membrane. So
yeah, we also trying to visualize FTSA, but it doesn't work yet. So I hope we will see that.
Let's see. We will also working on that part.
Yeah, thank you very much.
Thank you.